At the University of Arizona Bio5 Institute, we are focused on tackling complex challenges such as disease, hunger, water and food safety, and other health and environmental issues facing our families, communities, and the world. Bio5 brings together hundreds of multifaceted experts that include world-class scientists, engineers, physicians, and computational researchers in a team science environment designed to creatively solve difficult problems. This approach has resulted in disease prevention strategies, promising new therapies, innovative diagnostics and devices, and improved food crops. Join us each week as we talk about science with researchers, staff, and students from the University of Arizona's Bio5 Institute.
Hello, welcome to another episode of Science Talks, a conversation hosted by the University of Arizona's Bio5 Institute. I'm your host, Dr. Brittany Ulorn. Bacteria were among the first forms of life on Earth, and these microscopic organisms continue to rule our world today. While some strains cause infection or spoil food, others are essential to providing nutrients to plants, fermenting foods, and supporting our gut and reproductive health. Today, we're talking with Dr. Melissa Herbst-Kralovetz, Associate Professor of Basic Medical Sciences, as well as Obstetrics and Gynecology at the College of Medicine, Phoenix. She's also the director of the Women's Health Microbiome Initiative. Dr. Herbst-Kralovetz specializes in the area of host microbiome interactions of the female reproductive tract to ultimately develop better diagnostics, preventatives, and treatments for things like gynecological cancer. Thank you so much for joining us today.
Thank you for having me. I appreciate the opportunity to chat with you today.
Of course, yeah, I'm very excited about it. So I think bacteria often gets a bad rap. When people hear about bacteria, especially when they find out that bacteria live inside of us, they might get a little concerned. So talk to us about this delicate balance between good and bad bacteria, and particularly how it relates to your work with the female reproductive tract.
Sure. So I agree. Most people hear bacteria and then they think bad and they think, let's kill it with antibiotics. And, that's worked well for us for a while, but now we're gaining a better appreciation for actually the good bacteria that contribute to our overall health and well-being. And so we're learning a lot about that from our gut, and a lot of people that study the gut microbiome. In terms of the female reproductive tract and what we study, we've known since the 1800s that these bugs called lactobacilli actually contribute to women's health. But we're gaining, again, a further appreciation for this. And these lactobacilli are actually found in the gut as well, but they come in different flavors in the female reproductive tract. And we know that similar to the gut, these lactobacilli have protective mechanisms. So they contribute to health and homeostasis within the female reproductive tract. And we know that if you deplete these bacteria, or if they become destroyed or by particular compounds that you might use, feminine hygiene products, etc. And we know that these bacteria get depleted, then that's an opportunity for what we call the bad bacteria to overgrow and then actually contribute to a whole host of adverse women's health outcomes.
Fascinating. Yeah, it does seem like a delicate balance, and it can be altered by the things we consume. I'm just interested, where did these bugs come from in the first place? Are we born with them? Does it come from the things we eat, from the environment? How do they get inside of us?
Yeah, so that's a complex question that a lot of people are dissecting in terms of the origins of the microbiome, at what is the first kind of exposure that we have to these microbiota. And, the jury is still out. That's a highly controversial topic. Actually, you might not be aware, but it is a controversial topic. But what we know is that when you have these lactobacilli, again, that it does change across a woman's lifespan. So when you're born, you have these lactobacilli because you actually have estrogen from your mom. And then that goes down soon after birth. And during adolescence, you have more of these anaerobic organisms and not as many lactobacilli. But as you enter into that menarcheal phase and you're premenopausal, you have a lot of estrogen. That estrogen feeds the glycogen in our epithelium, and we have more lactobacilli. And so during that entire phase of the lifespan, lactobacilli dominance is again associated with overall vaginal health. During menopause, when that estrogen is depleted, those glycogen levels go down, that epithelium may change, and we may not have as much lactobacilli dominance during that time, which can cause a lot of issues. If we give back hormone replacement therapy, then we can get back that glycogen and that food source for those lactobacilli, and we have more lactobacilli dominance again. So this lactobacilli dominance really does contribute to health and homeostasis throughout the lifespan, and that does change.
It's fascinating. I had no idea that the female reproductive hormones were influencing the bacteria in our reproductive tract. So
absolutely, it could impact our gut microbiome too. I think we're just starting to learn more about that.
Yeah, fascinating. So I know you mentioned that lactobacilli, the presence of them, is more protective, or it's a sign of good health, reproductive tract health. So tell me how losing that type of bacteria would impact different infections, maybe increase susceptibility to infection, or just increase disease in general.
Yeah, great question. So yeah, as I mentioned, there are different factors that can drive whether you have this lactobacilli depletion, and then you have this overgrowth of these bad bacteria or anaerobic organisms. And that condition we call bacterial vaginosis, or vaginal dysbiosis, so BV. And BV is really an enigma. It's very complex. It's what we call a polymicrobial condition. So it's not one single etiologic agent or pathogen that we're studying. So it makes it way more complex. And actually, I wanted nothing to do with it when I had a postdoc that started in my lab that was really interested in it. And then I got hooked, just like she got hooked, because it is really complex. And it is linked to so many of these adverse women's health outcomes. So what we're trying to do, and what we started doing originally, was really taking a reductionist approach to this very complex condition, because we didn't know what any of those bugs were doing. So it's a polymicrobial condition, but no one really had started to study and dissect those mechanisms of the bacteria that made up this polymicrobial consortium. And so what we're learning is that there's early colonizers, so bacteria that come in at an early stage and might contribute to this biofilm. And then we have secondary colonizers that come in later after that biofilm's been established. And those secondary colonizers have different mechanisms of action. So those early colonizers, we are hypothesizing, can contribute to that biofilm. And so they're using different mechanisms. And then you have these secondary colonizers that come in and colonize that biofilm. And those are the bugs that are driving things like inflammation, genital inflammation. And we know inflammation's bad, right? It's bad for a lot of things. It can be good. But in terms of promoting cancer and things like that, inflammation is bad. And that's how we're starting to kind of put together a picture of what are the functional roles of these individual bacteria. And then when you put them together, does that pathogenicity of those organisms change? So are there microbe interactions that are occurring that make them even more pathogenic? Or virulent, and contribute to some of these pathophysiological changes within that cervical vaginal microenvironment.
Wow.
Does that answer your question?
Yes, it does, but I have a follow-up question. So by doing the work in your lab and understanding the pathogenicity and by understanding the interactions between the different microbes and our bodies, how can your work then help to inform prevention or treatment strategies for these different diseases?
Yeah, that's a great question. So we have a couple different kind of arms of our research program looking at that, but overall, related to everything that I just mentioned in terms of the functional role of these individual organisms, if we want to target them, and we're still learning, we have so many groups working on this, which is amazing, to try to better understand what are the key bacteria that are driving this dysbiosis, and can we disassemble, for example, the biofilm by targeting some of these bacteria that contribute to the biofilm by disrupting that biofilm, for example, and then as I mentioned with the secondary colonizers, blocking some of those to block inflammation, for example. So we're still kind of in early stages, better understanding each of these functions. The other aspect of this is if we know that there's a microbial signature, or that some of these functions, some of the things that we're looking at too are, doing the immunoproteomics analysis and the metabolomics analysis, and potentially using those as markers for diagnostics for some of these conditions as well. And again, we're in the early stages there, but that's where we want to see this kind of translated back to the clinic so we can impact women's health in a positive way.
Definitely. Yeah, it sounds like this research, you'll be able to understanding the fundamentals, and then it has so many different avenues through which it can impact actual patients in the clinic. So I think that's incredible.
Mm-hmm.
I'm curious, so I know you mentioned a postdoc kind of brought you into the BV world, bacterial vaginosis world, but where did your original passion for doing this type of research stem from?
Yeah, so in terms of infectious diseases in general, it was really my undergraduate research experience. In Colorado, in Grand Junction, I started doing an undergraduate research project with Dr. Schounts up there. And we were working on the Sonombre virus at the time. So this was in the late 90s, early 2000s. And, that really got me geeked up. I originally wanted to go to medical school, and, doing that undergraduate research project got me hooked on infectious diseases. And the cool thing about Sonombre virus is that it doesn't cause infection. Well, it infects, but it doesn't cause disease in deer mice, but it does in humans, right? So that kind of differential host response, and what were those host factors or those immunologic mechanisms that cause these pathogens to manifest disease or not in different ways. That really was the first exposure to that and intrigue. And it's really kind of crossed over for my whole career, my graduate training and beyond, really just intrigued by all of these pathogens. And so I may not kind of stay in my lane and just work on one pathogen my entire life, because I'm just so intrigued by how each of these pathogens work differently. And so it really feeds into this whole question of BV and it being a polymicrobial condition, because I don't have to study just one single pathogen. So it feeds into that for sure.
Definitely. Yes, it's opened a lot of doors, both now and in the future for years to come for you as a researcher. And I love how you mentioned too that your undergraduate research experience is kind of what changed your mind from med school to pursuing grad school and now being a research faculty. So I think I had a similar experience where I went into undergrad thinking I'd be a pharmacist, and then I got into a research lab and I got my hands dirty and I became fascinated with all things science and research. So I think a lot of people can relate to that.
Yeah, absolutely. So important to have undergrads in the lab and provide them those experiences. And I definitely feel I need to pay it forward in that regard because it definitely changed my whole career trajectory.
Yeah. Tell me about the makeup of your lab. I know you mentioned you have a postdoc, but do you have grad students and undergrads currently working with you?
Absolutely. Yeah. I'm really grateful to have kind of all levels represented in the lab. I usually take on a high school student. Our high school program is not ongoing this summer due to COVID, but I usually have a high school student every summer. I take on numerous undergrads. And through different universities. So I have undergrads from other local universities as well as Bath University. So we participate in an exchange program with the UK. And that has been incredibly valuable where they come for a one-year placement in the lab. So undergrads, and then I've had graduate students, as well as the postdocs that you mentioned, but I also have the great opportunity to interface with our OBGYN residents and fellows. So that's a great experience as well. So I really get that entire spectrum of training, and I have to adapt for sure to those different levels and the programs that I'm involved with.
Right, most definitely. High school students are very different than your undergrads and grad students, postdocs, and even your residents. So, but I can see how not only are they benefiting from you and getting your mentorship, but I'm sure that you as a mentor benefit, and I'm sure the research also grows, getting both the academic perspective and that more clinical perspective from your mentees.
Absolutely. That's how we're able to translate findings and actually come up with new ideas because they're kind of the boots on the ground. They're in the clinic every day. They're seeing the problems that they want to tackle too. And so it is this reciprocal process that is so fun. And I'm so grateful to be a part of a med school and to be able to interface with our clinical collaborators and trainees.
Right. It's a great opportunity.
Yeah.
So kind of speaking about trainees and postdocs, so we've had two Bio5 postdocs, Dr. Robert Jackson in Dr. Conrad Van Dorslayer's lab, and then also Dr. Kate Rhodes in Dr. Maggie So's lab on our podcast. Mm-hmm. So both are collaborating with you using this novel 3D bioreactor technology to make lifelike representations of the female reproductive tract to enhance their research. So tell me, how have the connections that you've made through Bio5 enabled both you and your colleagues to make scientific advancements and discoveries that might not have otherwise been possible?
Absolutely. So going back to some of the things that I mentioned early on in terms of bacterial vaginosis and in this dysbiosis kind of setting you up for all kinds of, poor women's health outcomes, one of those is an increased risk of all sexually transmitted infections. So all viral, bacterial, as well as parasitic. And so, and this is why it's a huge public health problem, right? And so we're really interested in, okay, I mentioned some of the functional changes that occur with these bacteria that set up for not only a favorable environment if we're talking about lactobacilli, but this higher risk or higher susceptibility microenvironment. And that's really where Dr. Jackson and Dr. Rhodes come in. So they both study highly relevant sexually transmitted infections, right? So Dr. Jackson with HPV and Dr. Rhodes with Neisseria gonorrhea. And so with their help and their PI's help, Dr. Van Dorsler and Maggie So, we're able to start to better understand what are those factors that are driving that increased risk of infection, as well as growth and persistence of those organisms. And we can use our human three-dimensional models to study those interactions. So we're really excited about those opportunities to partner with them through our Bio5 collaboration to extend our findings, to make a more of an impact and understanding on those risk factors associated with those other infections, because most of those studies have been epidemiologic studies. And so we want to understand, is this something that's mechanistic? Is it, what is it about the microenvironment that might be creating more of a favorable, STI favorable environment?
Right. Well, I think that also goes back to what you were saying earlier about how you personally don't have to study just one pathogen for the rest of your life. Not only does your research open you up to so many doors, but then it allows you to have collaborations with people studying, viruses, different viral infections, bacterial infections. So I think just the nature of your work is so powerful and so broad that it can reach all these different avenues.
Yeah, it's definitely fun partnering with others and collaborating. Yeah,
I'm sure. So tell me more about the Women's Health Microbiome Initiative. So we mentioned that you're the director of that. So what exactly is this initiative and how is it helping to advance women's health?
So it goes back to some of the other questions that you had, and some of the translational aspects of our research program. So the Women's Health Research Program and Microbiome Initiative is really aimed at bringing together these clinical collaborators along with scientists, so bacteriologists, virologists, immunologists, as well as biostatisticians and bioinformatics gurus, to really start to look at not only the microbiome, but these multiomics analyses, and see, what is it about, again, the changes in the microenvironment that might be setting up for all of these disease outcomes. And so being able to really partner at this larger level, being able to have access to clinical specimens and to perform this translational research allows us to tackle some of these really complex women's health questions that are remaining. So we talked about some of those with regard to STI acquisition, but also gynecologic cancer. So we're really interested in studying cervical cancer and endometrial cancer, as well as cancer health disparities that are disproportionately impacting women here in Arizona. So our Latina population, as well as our indigenous populations, in addition to other questions like infertility and endometriosis. So we're really able to partner and do things at a bigger level through this initiative and through this program. And we've been able to build a kind of pipeline of clinical specimens, working with our clinical research team as well. So it's really exciting. It's an exciting initiative, and we're hoping to make some great discoveries.
Most definitely. I think you raise a really good point that not only does your work and your field require all of these different minds, but I think we're starting to see, and we've been seeing for a while, that the biggest questions in STEM, whether that's about diseases or food sustainability, they can't be solved by just one lab or one discipline. It really requires this collaboration of scientists and clinicians across disciplines to bring together their resources, both material, literal, and intellectual, to answer these big problems. So that's great that you're able to head this initiative to tackle these big questions in women's health.
Yeah, it's really fun bringing together all of these different groups, and I get great satisfaction out of doing that. So yeah, team science is definitely the way to go. We can't make these big, at least, the whole goal is to have high translational value in this research so that we can go back to the clinic and make an impact.
Right, definitely. So you are obviously incredibly busy and both successful in your work at the university, but I'm curious how you spend your time outside of lab. So how do you bring in a good work-life balance? What are some of your favorite hobbies, and how do they help benefit your career and your personal life?
Yeah, so I think we can't just be in the lab all the time. So, and I say I'm a recovering workaholic. So, it's always hard. I don't know that balance really exists. I think if anyone says they have it figured out, then they might be lying. I think it's, time is very personal, but it's so important that we spend that time rejuvenating and recharging. And the way that I do that is with my family. I have two boys. I have a 10-year-old and a 13-year-old, and they like to get dirty. They like to go outside, and we love camping as a family, doing outdoor activities. And I think it's so important to, reconnect with nature. And so that we can be recharged, so that when we come back to the lab, we have new questions, and we feel rejuvenated, and we can, really make these scientific discoveries and breakthroughs. So I don't know that balance exists, but I try to be able to spend time with my family and recharge in nature is the way that we like to do that.
Yeah, it's always a work in progress, that's for sure. And it totally resonates with me what you said about nature. That was one of the things that I love about Tucson, and that drew me there initially back when I was looking at a place to go for my undergraduate. It was just the mountains and the ability to escape the city, escape the town, and just be alone or be quiet or be with, the ones that I love, and kind of get rejuvenated and inspired. And it always helped me come back to school or the lab or now my job, just with a clearer mind and able to be more creative and productive. So that really resonates with me.
Yeah, absolutely. And Arizona is beautiful. Our desert is so beautiful. I'm grateful to be here. I grew up here in Arizona, and camping and getting out in the middle of nowhere. And, I've lived in other places in the US, including Texas, which it's a little bit harder to find those open spaces. So very grateful for that and our beautiful desert.
Agreed. Oh, do you have any upcoming camping trips planned?
We just did a camping trip about a week and a half ago. So, but we like to camp all year round, and especially in the fall, when it's better temperatures for camping.
A little warm right now. You have to go quite north, but
Absolutely.
We'll get out of the heat hopefully sooner rather than later.
Yeah, it's raining here today, so that's kind of a miracle. We need it.
I know. Doesn't happen often. Yeah. Well, I guess now that it is monsoon season, but can't take those times for granted.
Absolutely.
Oh, well, thank you again so much for joining us today. It was wonderful to hear more about your research, where your passion stems from, the different broad impacts that it has, and also just a little bit about you personally.
Yeah, thank you for having me. You did a fantastic job too. Oh, yeah, with the follow-up questions and everything. So I appreciate that.
Oh, thank you. I appreciate it. It's easy to have a conversation with someone like yourself that is so engaging and makes me think of even more questions, makes me want to learn more. So I hope our listeners feel the same way.
Me too.
Aw. Well, thank you to our listeners for joining us for another episode of Science Talks. For more information, please visit our website at bio5.org. And from all of us at the Bio5 Institute, we'll see you in the next episode.
To our audience for tuning in to another episode of Science Talks. Continue the conversation with us next time as we learn more about the science happening at the University of Arizona's Bio5 Institute.